Okay, let's unpack this. Imagine a future where, I don't know, a common infection could suddenly become untreatable, where even a simple cut might actually turn fatal. Sounds like science fiction, maybe.
Right, but it's really not.
Yeah, this isn't some dystopian story. It's a critical looming threat that's unfolding right now globally. So today we're taking a deep dive into bacterial antibiotic resistance. We're going to shine a light on the most dangerous pathogens, the ones posing the gravest risks to human health right now.
And our mission for you today is pretty clear, I think. We want to cut through the noise, really sift through the scientific sources, and help you understand the current state of antimicrobial resistance, or AMR. We'll look into the ingenious ways bacteria manage to evade our drugs, and critically, the specific dangers from some key pathogens the World Health Organization has flagged as, frankly, top priority.
Exactly. And this deep dive, it's your shortcut to being really well informed on what is a true global health emergency. We're hoping to uncover some surprising facts, some crucial insights without, drowning you in jargon. For instance, get this. At least 700,000 people worldwide die each year because of AMR. That's already happening. And the WHO predicts that number could just skyrocket to a staggering 10 million annually by 2050 if we don't get new treatments sorted.
It's an urgent wake-up call, isn't it?
It absolutely is a massive challenge.
Now think back to that sort of aha moment with antibiotics. They really did revolutionize medicine.
Oh, completely.
Before these wonder drugs, bacterial infections like meningitis or bloodstream infections, bacteremia, they were often basically death sentences.
That's right.
And then suddenly they became treatable. It just utterly transformed healthcare. It did. But what's truly fascinating here, and maybe a bit scary, is that resistance isn't some new trick bacteria learned recently. They've been evolving defense mechanisms for eons. But our actions have drastically sped things up. We've turned a natural evolutionary process into, an acute public health emergency.
Okay, so what actions are we talking about? What's really driving this acceleration?
Well, what's kicking this problem into high gear? It boils down to a few big things. First, just the sheer overuse and misuse of antibiotics. We're using way too many, not just in human medicine, but maybe even more significantly in farming and animal agriculture. That's a huge factor.
Right, and it's not just the drugs themselves.
No, there are also broader socioeconomic issues, things like poor community sanitation, unsafe food handling practices, and sometimes inadequate infection control, even in hospitals and clinics. And finally, these antibiotic compounds, they're ending up in our environment. They accumulate in soil, in water, creating this constant low-level pressure for resistance to develop.
Wow. And you mentioned historical context earlier. That example about Staphylococcus aureus resisting penicillin back in the late 1950s, that's really eye-opening.
It is, isn't it?
It just shows this isn't a brand new problem. And I suppose for a while we got maybe a bit complacent. New drugs came along, like vancomycin and methicillin in the 60s.
Exactly. It suggested maybe we could just keep inventing our way out of trouble.
But the bacteria didn't cooperate. They quickly developed new resistance mechanisms, showing just how relentless and adaptable they really are.
And that adaptability, that relentless pressure, that's precisely why the WHO formally identified antimicrobial resistance as one of the top 10 global health threats back in 2019. It's an ongoing severe impact on human well-being. Make no mistake.
So let's be crystal clear. When we talk about antimicrobial resistance, what does that actually mean for us? For people. It's not like our bodies become resistant.
Yeah, that's a really important distinction. No, it's not us. The World Health Organization defines it simply as when microorganisms, bacteria, fungi, viruses, parasites no longer respond to the medicines, the antimicrobials, that used to effectively treat the infections they cause. So it's the microbes themselves, the bacteria in this case, that adapt and change.
Got it. The bugs learn the tricks. So how do they do that? How do bacteria actually become resistant?
Right. They achieve this resistance primarily through two main pathways. First, there's what we call natural resistance. This can be intrinsic, which just means a particular bacterial species is naturally built, maybe structurally, to resist certain antibiotic classes, even if it's never encountered them before.
Well, it just doesn't have the target the drug goes after.
Or the drug can't get in. Exactly. For example, Pseudomonas aeruginosa naturally shrugs off certain types of penicillins and cephalosporins, which is part of its makeup. Then within natural resistance, there's also induced resistance. This is where a bacterium has the genes for resistance, but they only get switched on or activated when it's actually exposed to clinical amounts of an antibiotic.
Okay, so natural can be built in or switched on. What's the other pathway?
The second main type, and this is often the one that causes more widespread problems, is acquired resistance. This is where bacteria actively learn to resist picking up new defenses. And this happens mainly through two distinct processes. One is simple mutation, just random changes, typos really, in the bacterial DNA when it copies itself. If one of those random changes happens to help the bacterium survive an antibiotic,
It gets passed down to its offspring. Survival of the fittest, microscale.
precisely, vertical transmission. The other process, and this is arguably much more dangerous for spreading resistance quickly, is DNA transfer, often called horizontal gene transfer or HGT.
Horizontal, meaning not parent to child.
Exactly. This is like bacteria sharing their cheat sheets, their resistance genes, directly with each other, even if they're not related. And they have some incredibly clever ways of doing this. They can perform transformation, which is basically picking up stray bits of DNA encoding resistance that might be floating around in their environment, maybe from dead bacteria, almost like scavenging useful code.
Wow.
Or there's transduction, where a virus that infects bacteria, a bacteriophage, accidentally packages up some resistance DNA from one bacterium and then delivers it to another when it infects the next one, like a tiny, unwitting courier.
That's amazing.
And then there's conjugation. This is probably the most direct way. It's often described as bacterial mating. One bacterium forms a temporary bridge, like a little tube, to another and directly transfers a copy of its resistance genes, often on a mobile piece of DNA called a plasmid.
So they're literally sharing the blueprints for resistance.
They are, and the critical consequence here is that these antibiotic-resistant genes can spread incredibly rapidly through a bacterial population, turning previously susceptible bacteria into resistant ones almost overnight. It fuels the rapid rise of multi-drug resistance.
It's truly amazing the diversity of strategies these microscopic organisms use to outsmart our best drugs.
Yeah.
When you dig into the science, it really is like a masterclass in biological trickery. Can you walk us through some of their cleverest tactics at the cellular level?
Absolutely. And the bacteria's tactics often directly counter exactly how our antibiotics are designed to work. It's a real arms race. For instance, a huge class of antibiotics, the lactams that includes things like penicillins, cephalosporins, carbapenems, they work by messing up the bacterium's ability to build its cell wall, crucial for their survival.
Right, like taking bricks out of their wall.
Yeah. Kind of, yeah. But bacteria fight back by producing enzymes called alactomases. Think of these like molecular scissors. They literally find the antibiotic molecule and cut a specific part of it in activating it. And there are many types, including some really problematic ones like ESBLs, extended spectrum beta-lactamases, and carbapenemases, which can dismantle even our most powerful last resort Volactam drugs.
Okay, so they disarm the weapon. What else?
Then you have antibiotics that target protein synthesis inside the bacterial cell drugs like aminoglycosides, macrolides, or tetracyclines. Proteins are essential for everything the cell does. Here, bacteria have multiple tricks. They might use enzymes to chemically modify the antibiotic itself, making it useless. Or they can subtly alter the target site, the part of the cell's machinery the antibiotic normally binds to, so the drug just can't lash on effectively anymore. It's like changing the lock so the key doesn't fit. And another big one here is activating efflux pumps. We mentioned these briefly. They're like tiny pumps or pores in the bacterial cell membrane that actively recognize the antibiotic once it gets inside and just pump it right back out before it reaches a high enough concentration to do damage.
The bouncer analogy again, just kick it out.
Exactly. Keep the concentration inside low. Then for drugs that inhibit DNA replication, like fluoroquinolones, bacteria often develop mutations in the target genes within their own DNA. These mutations change the shape of the proteins the drug interacts with, making it harder for the antibiotic to interfere with DNA copying.
Okay.
And finally, for drugs like sulfonamides and trimethoprim, which block the production of folic acid, something bacteria need to make DNA and RNA resistance often spreads through that horizontal gene transfer we talked about. They acquire genes for alternative enzymes that are insensitive to the drug, allowing them to just bypass the blockage.
It sounds like they have a whole toolkit. If we were to step back and categorize, what are the main overarching strategies?
Yeah, if we boil it all down, these incredibly diverse mechanisms broadly fall into four main categories, four overarching strategies. 1. Decreased drug uptake. They make it harder for the antibiotic to even get inside the cell in the first place, maybe by altering the pores in their outer membrane. 2. Drug target alteration. They change the specific part of the cell that the antibiotic is designed to attack, so the drug can't bind or work properly. 3. Drug inactivation. They produce enzymes like those balactamases that literally destroy or modify the antibiotic molecule itself. And 4. Drug efflux pump activation. They actively pump the antibiotic out of the cell. They're incredibly resourceful, always finding a way around our defenses.
That's quite the arsenal. Are all these mechanisms equally concerning, though? Or are some, like, truly more problematic when it comes to developing new drugs
Right.
or seeing resistance spread really fast?
That's a good question. They're all concerning, obviously, because they render treatments ineffective. But I'd say the ability to acquire new resistance genes, particularly through that horizontal gene transfer conjugation transduction transformation, that's especially problematic. Why? Because it means a brand new resistance mechanism doesn't have to slowly evolve through mutation in just one bacterial lineage. Instead, a resistance gene that pops up somewhere can spread like wildfire through different bacteria, even different species sometimes. It accelerates the whole process dramatically.
That makes perfect sense. It's like sharing the winning lottery ticket instead of everyone having to buy their own.
Kind of, yeah. It allows for very rapid dissemination of resistance.
Okay, so understanding that rapid spread is key. Let's bring in the WHO again. In 2017, they released this global priority pathogens list. They categorized bacteria based on how urgently we need new antibiotics for them. Critical, high, and medium priority. Today, we're really zeroing in on that critical priority group. These are the multi-drug resistant bad guys that are particularly dangerous, especially for hospitalized patients, people relying on medical devices like ventilators or catheters, basically the most vulnerable.
That's right. These are the top tier threats.
And these critical pathogens conveniently have a memorable acronym, ESCAPE, ESKAPE. That stands for Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumonia, Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacter species. And the name fits, right? They're called ESCAPE because they've become incredibly adept at, well, escaping the effects of almost all our available antibiotics.
It's a fitting name, unfortunately. So let's maybe delve into each of these escape pathogens a bit, just to highlight their unique resistance challenges. Where should we start?
How about Acinetobacter baumannii? It sounds like a real hospital nightmare.
It certainly can be. So Acinetobacter baumannii, or A. baumannii, it's an aerobic gram-negative bacterium. It's what we call an opportunistic pathogen. It doesn't usually harm healthy people, but it thrives in hospital environments and can cause really serious infections in vulnerable patients. Pneumonia, bloodstream infections, wound infections. Its resistance profile is formidable. Why? Because it's a master of using multiple strategies simultaneously. It's incredibly good at producing a huge array of those O-lactamase enzymes we discussed. It makes all four major classes, often getting the genes through that horizontal transfer. This includes enzymes that destroy carbapenems, are really potent drugs.
So it just chews up our best antibiotics.
Pretty much. And on top of that, it uses many different types of efflux pumps to just flush various antibiotics out of its system. Aminoglycosides, tetracyclines, fluoroquinolones, even lactams sometimes. Overexpression of certain pumps is linked to resistance against even newer drugs like typecycline. It can also modify its outer membrane pores to reduce drug entry and even alter the targets of antibiotics, like the proteins involved in cell wall building or DNA replication. It throws everything at us.
Which must make treatment incredibly difficult.
Extremely. Carbapenems used to be reliable, then maybe drugs like minocycline or tigecycline, but resistance to those is now significant. Clinicians are often forced to use combinations of drugs, sometimes resorting to older, more toxic antibiotics like colistin as a last resort, often paired with something else. But here's the kicker. Resistance even to these multi-drug combinations is increasing. It leaves doctors with frighteningly few good options sometimes.
Wow. Okay, so A. baumannii is a major hospital threat, really weaponized with enzymes and pumps. Stepping slightly outside the purely hospital setting, maybe, we often encounter another equally concerning bug, Pseudomonas aeruginosa. What makes that one such a persistent problem?
Right. Pseudomonas aeruginosa, or P. aeruginosa, also an aerobic gram-negative bacterium. This one's actually pretty common in the environment soil, water, but it's a leading cause of severe infections, especially nosocomial, hospital-acquired ones, in immunocompromised patients. Think severe respiratory infections, particularly in patients on ventilators, bloodstream infections, serious burn wound infections. What makes it tricky is its blend of intrinsic resistance. It's naturally tough to begin with, partly due to its outer membrane, plus its uncanny ability to quickly acquire new resistance genes.
So it starts tough and learns fast.
Exactly. It's excellent at overexpressing those efflux pumps we talked about, kicking drugs out. It's also good at decreasing the permeability of its outer membrane, essentially putting up a tougher shield so fewer drugs get in. And like A. baumannii, it can acquire or mutate genes for resistance, affecting antibiotic targets. And it produces a broad spectrum of lactameses, including its own inducible one called AMP-C, plus others it acquires that can destroy carbapenems. It also has specific enzymes to neutralize aminoglycoside antibiotics.
So similar strategies, but maybe a different niche sometimes.
Somewhat, yes. Both are huge problems in healthcare settings. Treating Pseudomonas also often requires strong antibiotic combinations, maybe colistin paired with specific anti-pseudomonal drugs like certain penicillins, cephalosporins, or carbapenems. Sometimes combinations involving fosfomycin are used. But again, the bacteria are so adaptable that even these multi-drug approaches face increasing failure rates.
Okay, let's switch gears slightly. Gram-positive now. Staphylococcus aureus, or S. aureus, this is probably more familiar to people, causes everything from relatively minor skin infections, boils, to really life-threatening conditions like endocarditis or sepsis.
Absolutely. S. aureus is a major human pathogen, gram-positive, facultative anaerobe, meaning it can live with or without oxygen. It's highly contagious, found on skin and in the nose of many healthy people, but what always stands out is its incredible, almost notorious ability to develop resistance, seemingly in real time with our drug development.
Yeah, the history here is striking, isn't it?
It really is. Penicillin was discovered, became widely used, and boom, within just a few years, penicillin-resistant S. aureus, producing those wool lactamase enzymes, appeared. Then methicillin was developed specifically to overcome that initial resistance.
And then MRSA showed up just 2 years later.
Exactly. MRSA, methicillin-resistant S. aureus, first identified in 1961. MRSA's main trick is acquiring a gene cassette, usually a MEK-A or MEK-A, through horizontal gene transfer. This allows it to produce an alternative penicillin-binding protein, PBP2A, which lactam antibiotics like methicillin just can't bind to effectively, so the drugs are useless.
And MRSA became a huge problem hospital and community acquired. For years, vancomycin was the go-to drug for serious MRSA infections, right? The last line of defense.
It was. Vancomycin works differently, targeting cell wall synthesis in another way, but then resistance started appearing there too. Interestingly, it first showed up significantly in another type of gram-positive bacteria, Enterococci, we'll get to them creating VRE, vancomycin-resistant Enterococci, and then through what seems to be a transfer of those resistance genes from VRE, we started seeing VRSA, vancomycin-resistant S. aureus, in recent years. It's still relatively rare compared to MRSA, but incredibly worrying.
How does that vancomycin resistance work?
It's clever. S. aureus acquires a set of genes, often the VANA operon on a mobile piece of DNA called TN1546, likely from VRE. These genes allow the bacteria to change the very structure of their cell wall precursors, changing a D-alanine end group to D-alanyl D-lactate. Vancomycin normally binds tightly to the first structure, but binds very weakly to the modified one, so the drug loses its grip. We also see VISA, vancomycin intermediate S. aureus, which shows reduced susceptibility but isn't fully resistant like VRSA. It seems to have a different, more complex mechanism, often involving thickening of the cell wall.
And beyond MRSA and VRSA, staph has other tricks too.
Oh yes. It can readily mutate its target genes to become resistant to other classes like fluoroquinolones. It can overexpress efflux pumps, like the Nora pump. Because resistance rates can be high, especially for skin infections, combination therapy is often recommended, using drugs like fusidic acid or rifampicin, but resistance develops to those too if used alone. There's also a class of drugs called MLS agents, macrolides, lincosamides, streptogrammins. Clindamycin is a common lincosamide used for staph, but resistance is increasing and sometimes it's inducible, meaning the bacteria only show resistance when exposed to the drug. There's a lab test called the D test, which is crucial to detect this hidden, inducible clindamycin resistance. Otherwise, treatment might fail unexpectedly.
Fascinating. Okay, next on the escape list. Klebsiella pneumonia or K pneumonia, gram-negative, part of the Enterobacterales family, like E. coli. What's its story?
K pneumonia is a significant cause of both hospital-acquired and community-acquired infections. Think UTIs, pneumonia, even serious liver abscesses and bloodstream infections, especially hitting immunocompromised patients hard. It spreads pretty easily from person to person or via contaminated environments. Its main claim to infamy in the resistance world is that it's become a major hub, a collector, for acquiring genes that encode those powerful bolus stomaces we keep mentioning, particularly ESBLs, and, critically, carbapenemases.
Carbapenemases again, those seem to be a recurring nightmare.
They really are. Carbapenem-resistant K pneumonia, often shortened to CRKP, represents a massive global health threat. Strains carrying certain carbapenemase genes, like the notorious black PC gene, Klebsiella pneumonia carbapenemus, are resistant to nearly all lactam antibiotics, including the carbapenems, which are often our true last-line drugs for serious gram-negative infections. Treating CRKP infections is incredibly challenging. Options are limited, often relying on older, more toxic drugs like colistin or tigecycline or sometimes difficult combination therapies. The outcomes for patients with CRKP infections can be very poor.
Scary stuff. Okay, moving along the list. Enterobacter species. This includes bugs like Enterobacter cloacae and what used to be Enterobacter aerogenes.
That's right. Klebsiella aerogenes now, actually. It got reclassified recently due to genetic similarity to Klebsiella. But yes, this group, Enterobacter spp, are motile aerobic gram-negative bacilli. Like Klebsiella and Acinetobacter, they're primarily opportunistic pathogens causing trouble in immunocompromised, often hospitalized patients. UTIs, respiratory infections, surgical site infections, their resistance story. They are incredibly adaptable and seem to acquire a huge range of resistance mechanisms. Many strains produce ESBLs, making them resistant to most penicillins and cephalosporins. And crucially, like Klebsiella, they are increasingly acquiring various carbapenemyces, including types like VIM, OXA, NDM, New Delhi metallobalactamase, and KPC. They also have a knack for permanently derepressing or overexpressing their own intrinsic AMC-balactamase, which adds another layer of resistance to many cephalosporins.
So another group hoarding carbapenemyces and other resistance genes. What does that mean for treatment?
It means multi-drug resistant strains are common, often resistant to almost all available standard drugs, except perhaps tigecycline and colistin. And even that's not guaranteed. What's deeply concerning is the emergence of pan-drug resistant strains, particularly in K aerogenes, showing resistance even to colistin. And a really tricky thing some of these strains can do is harbor undetectable subpopulations of colistin-resistant bacteria within a mainly susceptible population. This can lead to treatment failure, even if initial tests suggest susceptibility, complicating diagnostics and therapy choices even further.
It's pan-drug resistant. That's a terrifying term. Okay, last on the escape list. Enterococci, specifically mentioning Enterococcus faecium.
Yes, Enterococci. These are gram-positive cocci, facultative anaerobes, normally found as common residents in our gastrointestinal tract. Two main species cause human infections, Enterococcus faecalis and Enterococcus faecium. Historically, E. faecalis was considered more pathogenic, causing more infections. But E. faecium has emerged as a major problem because it is often much more resistant to antibiotics. It causes severe morbidity and mortality, particularly in immunocompromised hosts in hospitals, think catheter-associated UTIs, endocarditis, bacteremia.
So why has E. faecium become so resistant?
It's a combination of factors driving this rapid increase in resistance. First, the extensive use of broad-spectrum antibiotics in hospitals, like certain penicillins and especially cephalosporins, to which Enterococci are intrinsically somewhat resistant, actually selects for and promotes the colonization and overgrowth of resistant E. faecium. We basically clear out the competition for it. Second, Enterococci have significant intrinsic resistance to several common antibiotic classes. Third, they have an exceptional capacity to acquire and disseminate resistance determinants, those genes we talked about, often carried on mobile genetic elements like plasmids and transposons. They're great at sharing resistance.
And this links back to VRE, right? Vancomycin resistance.
Exactly. The emergence of VRE, vancomycin-resistant Enterococci, primarily E. faecium, in the 1990s was a huge turning point and a major wake-up call about acquired resistance in gram-positives. As we touched on with VRSA, vancomycin resistance in VRE is often mediated by acquired gene clusters, most commonly the VAN-A gene cluster carried on that mobile transposon TN1546. This fundamentally changes the drug's target in the cell wall precursor, that DLL to TLDLX switch, making vancomycin ineffective.
And it's not just vancomycin.
Oh no. Many VRE strains are also multi-drug resistant. They have intrinsic resistance to lower levels of aminoglycosides like gentamicin to bromomycin, but can acquire genes for enzymes, AMEs, aminoglycoside-modifying enzymes, that confer high-level resistance, knocking out synergistic combinations with cell wall active agents. High-level resistance to fluoroquinolones is also common, usually linked to point mutations in their DNA gyrase and topoisomerase genes. Gyra and Park, and sometimes efflux pumps like Nora play a role too. Resistance to newer drugs like linezolid and daptomycin is also emerging. E. faecium is a truly challenging nosocomial pathogen due to this multi-drug resistance.
Yeah, okay, so we've run through the escape list. It's clear that while AMR is at its core a natural evolutionary process, our actions, antibiotic overuse, poor infection control, global travel have profoundly amplified it, leading to these really dire consequences with these specific pathogens.
Indeed. And it's worth mentioning it's not just these classic mechanisms we've detailed. The picture keeps getting more complex. There are a couple of other maybe less commonly discussed but important resistance concepts that further complicate this whole fight.
Oh, like what?
Well, one is something called heteroresistance, or HR. This is really fascinating and quite worrying. It describes a situation where, within a bacterial population that seems susceptible to an antibiotic based on standard lab tests, there's actually a small, pre-existing subpopulation of cells that are resistant.
So most of the bugs are sensitive, but a few are already resistant, hiding in the crowd.
Exactly. And this resistant subpopulation might be present at such a low frequency that our routine tests miss it. But when you then treat the infection with that antibiotic, you kill off all the susceptible cells.
And inadvertently select for that hidden resistant minority, which can then rapidly grow and take over, leading to treatment failure.
Precisely. It raises a really important question. If some bacteria are effectively hiding their resistance like this, how do we reliably diagnose and treat an infection if our standard susceptibility tests might be misleading? It adds a layer of uncertainty.
That is tricky. What's the other concept?
The second concept is the mutant prevention concentration, or MPC. We usually talk about the MIC, the minimum inhibitory concentration. That's the standard measure, the lowest concentration of an antibiotic needed to inhibit the visible growth of bacteria in a lab test.
Right, we hear about MICs a lot.
But the MPC is different. It's defined as the antibiotic concentration needed to prevent the growth of all bacterial mutants in a large population, including those rare, slightly less susceptible first-step mutants that might still be able to squeak by and grow at concentrations just above the MIC.
So MIC stops most growth, but MPC stops even the potential mutants from emerging.
That's the idea. It's a higher bar. Looking at the relationship between the MPC and the MPC-MIC ratio can give us insights into how likely an antibiotic is, at certain concentrations, to select for resistance during therapy. The goal would be to use dosing strategies that keep drug levels above the MPC for as long as possible to actively prevent those resistant mutants from ever getting a foothold. It's a more proactive way of thinking about dosing to suppress resistance emergence.
Okay, heteroresistance and mutant prevention concentration, two more layers to this already complex problem. So let's try and bring this together for you, the listener. What does this all really mean? The escape pathogens, as we've heard, represent these truly deadly bacteria. They have rapidly growing multi-drug resistant properties, and worryingly, their resistance strategies seem to be widely shared, jumping between these different species. It's a collective threat, isn't it?
It absolutely is. And the fight against AMR, against these escape bugs, is incredibly complex. It involves trying to manage natural bacterial evolution, which we can't stop, while simultaneously correcting the human actions that have accelerated it so dangerously. There really is no single silver bullet here.
So what's the path forward? To limit the spread of escape pathogens and antibiotic resistance more generally, it seems absolutely imperative that we get much more careful, more vigilant in surveillance tracking where resistance is emerging.
Yes, surveillance is critical.
Alongside that, really implementing strong antimicrobial stewardship programs. That means using antibiotics more wisely, only when necessary. The right drug, right dose, right duration.
Crucial. And that stewardship needs to happen not just in human health care, in hospitals and clinics, but just as importantly in agriculture, in food animal production, where huge volumes of antibiotics are still used.
It's not just a clinical problem then, is it? It's really a societal one, a one health problem that demands attention across different sectors.
Exactly. That stewardship piece is fundamental. And then, look into the future, that needs to be combined with continued efforts to develop brand new antibiotics, drugs with novel mechanisms that bacteria haven't seen before. Though that pipeline has been worryingly dry. And we also need to seriously invest in and explore alternative approaches, things like developing ways to inhibit biohelm formation, those protective slime layers bacteria form, or therapies that target virulence factors, making bacteria less harmful even if not killing them directly, and things like bacteriophage therapy using viruses that naturally prey on bacteria. These innovative strategies are key.
So better use of what we have, finding new drugs, and exploring totally different ways to fight bacteria.
That combination is really our main hope, our main strategy, to try and slow the global spread of these multi-drug resistant strains like the escape pathogens.
Okay, so maybe a final thought to leave you with. If these incredibly adaptable foes, these bacteria, have been honing their resistance skills for literally millennia, and yet we, through our actions, have accelerated this crisis in just a matter of decades, What responsibility do we all have, not just scientists and doctors, but everyone, in understanding and maybe influencing this global health crisis? And looking ahead, what truly innovative, maybe even unexpected approaches might finally turn the tide for good in this evolutionary arms race?